Okay, let's have a class, and we will do 2 more biochemistry tests, and then we're going to do antibiotic Kirby Beau test. So let's see what we have. So this is a biochemistry test 3. It should be lab 14. Okay, before we go talk about the new things, let's do some of the review. So what we have is, number 1, catalase test. And remember what we did, we had a bacteria on the neutron agar. The left side is E. coli, and right side is Staphylococcus aureus. This is neutron agar. So what are we going to do? What we're going to do the confirmation test. We will do is adding H2O2, because H2O2, if bacteria has a catalase, will break down, become water and oxygen. And this means bubbling. So we're going to do it all together. Okay? Let's say, can somebody give me agar plates? Do you have a sample? Just give one sample to me. Yeah, just give me one sample. And I need a Staphylococcus aureus, maybe this one, yeah. Okay, let's just do a demonstration. So I have H2O2, hydrogen peroxide. It's very simple. I'm going to dump them. Cup of drop. Can you see one side is you start to have bubbling? Well, E. coli certainly has it too. See the bubbling? See the bubble? You can see here, the camera. See the bubble? See the bubbling right here? See the bubble? Yeah? So when you add it, it will show you bubble. So that's, you will say, this is positive, certainly. This one will be light positive. Okay, slightly. Okay, that's catalase test. Second test, SIM test review. I want first to see, does anybody who has a slant which is black? That is black. That is black. Which bacteria you added? Citrobacter. Okay. This is sulfide. Okay, which bacteria positive? Citrobacter is positive because it's showing you black color. That is sulfide reduced to sulfide. Okay, now let's do a test. When you see the whole black color, does that means strong motility? Of course. So let's give an example. When you see this guy here, does this mean have a strong motility? Oh yes, because it goes everywhere. I want to have another one. Who have E. coli? Give me the E. coli. Yeah. Oh, one, just one. That one. Yeah. Okay, when you see E. coli, you don't see any change. So that's sulfide negative. And then you look at the middle. The line is clearly in the middle. You can see the line is clearly in the middle. Did you see it? That means no motility. That's what the E. coli looks like. This did a good job. I mean, so this tells you the motility is positive or negative. So that's what we have so far is, you see this guy is right stay in the middle. That is E. coli. So no motility. Okay, what's the last one we're missing? This is Indo. So how we do? We said that we need a reagent that is called Kovac reagent. So I need to find the Kovac reagent. Whether we had a Kovac reagent, we should have it somewhere. That's focus plus here. Methyl red. We have a Kovac Indo reagent. So we shake it a little bit. Let's see whether it's positive or negative. Sometimes these doesn't work. Let's get a E. coli. Okay, we're gonna add a little bit of drop on the top. Did that become cherry red? I don't know. We'll see. Go a couple of drops. Looks good. Maybe not good. Maybe good. Oh, start showing. Cherry red. See? That become cherry red. Look at it. How beautiful. Good? Okay. So always remember, what's the reagent we added here? KOVAC. It's called Kovac. And what's the color it is? Looks like cherry red. So that's good. So it is positive. E. coli, Indo, positive. You always remember. You can do it by yourself. Okay, now let's do this guy to see whether this is positive or not. Maybe Citrobacter is also positive. We don't know. Let's see. Looks like not. No change. This all have to be done like a 30 seconds a minute. You can't be putting it there whole day. Okay, if you put it whole day, maybe it become cherry red again because there's a bacteria in the environment or contamination. See, this doesn't have anything. Good job. That means didn't contaminate it. Couple of years teaching, last many years I see somebody have tubes everywhere is have E. coli because Indo positive. So that's good job. See, positive and negative. That can be an exam question. Which one is positive? Which one is negative? And what's the reagent we added is Kovac. And that's testing for what is Indo. What is Indo? Byproduct of tryptophan. Is that right? That's a whole story. So that looks good. Very good. I'll return to you. You can take a picture. Okay. And one is positive, one is negative. So SIM test we finished. Okay. The third one, what we did is phenylalanine deaminase test. So we said we have a slants. Looks like it's a very similar slants. Then what we did, we inoculate the bacteria when we assign the culture. So let's say we have a Citrobacter, and we also have a Bacillus, we have an E. coli. And what we're going to do, you're going to add a solution there, which is ferric chloride. And I have ferric chloride, I think. I need to find it. Where's the ferric chloride? So real, where is ferric chloride? They definitely have somewhere. I haven't seen here. We have a, should have ferric chloride. Okay, right here. So who has Citrobacter? Let's go here. You have a Citrobacter there? Yeah. Who has E. coli? You have E. coli? How about let's just Bacillus, okay? Is that Bacillus? Oh, which? Okay.
I have the same.
You have the same. Who has E. coli? Okay, just give it to me. Very good. What's it? Okay, we have E. coli and also we have Citrobacter. So let's do it. How we do it? Okay, then we're gonna add a reagent. So ferric chloride we're gonna add on the top. We have to see which one is contaminated. Maybe they're the same. Okay. Maybe the reagent is now looks good. Okay. So we'll see. So when we do that, we're gonna be doing like this. Is anyone showing you dark green color? Ideally, Citrobacter should be dark green color. But looks like the same. So that means that may not be a Citrobacter. See, if you line it deaminates, it didn't deaminate it. It's the same. Yeah, they didn't show dark green color. This one doesn't show. So let me see, we have another reagent. Maybe the reagent is old. Yeah, it's disappointed a little bit. Looks like there's no difference. See? That's not good. So it doesn't matter. Ideally, usually Citrobacter is positive, the other one is negative. So I'll just return. Get back to her. I don't know why, but I will find another sample for you. Okay, ideally, this one is positive, and this is negative. Citrobacter could be the deaminase test positive, but our results, that's our results, are both negative. Okay, so that's the question. Why? We don't know, maybe contamination or something. Okay, today we have 2 more testing we're gonna do. That's will be today's work. Okay, number 1. MRVP test. This test is simple. You have an MRVP solution, look like a neutron agar, and then you inoculate your assigned bacteria. The assigned bacteria will be stick onto E. coli or Enterobacter. Okay, why we have to do this test? This is a 2 ingredients there. MR is methyl red. This is used to test the acid products generated during fermentation. After fermentation. And what is VP? That's not a vice president, okay? Vogus Pruskia. Vogus Pruskia. That is neutral product after fermentation. Now, this testing is important. Why? It is a group test called IMVC testing. In the clinical, it's very important. What is I? Is Indo. We just talked about. What is M? Methyl red. I need to make sure this spelling is correct. METH, because I need to make sure this is correct spelling. Methyl red, it is METHYL. TH, not EL, YL. This V is VP, okay. Vogus Pruskia. C is citrate. This is a group of tests. Sometimes there's I. It doesn't matter. IMVC test. This testing to differentiate E. coli and the Enterobacter. So first of all, why we have to use this one to test it? Because E. coli and the Enterobacter looks very similar. They are gram-negative lods. Okay, non-endospore forming. And it is lactose fermentation positive. It is facultative. So these type of bacteria looks very similar. It's difficult to identify. So we have to use a biochemistry test. That is IMVC test. The results is magic. E. coli, positive, positive, negative, negative. Enterobacter, negative, negative, positive, positive. What did this looks like? A sticky ends. Is that right? Because go this way, goes that way, compromise with each other. That's why IMVC test is important. So today you're only going to do inoculation. On next week, we need to add a reagent A, B to do the confirmation test. Okay, so today you only inoculate E. coli or Enterobacter. It's very simple. You inoculate E. coli. And you bench partner inoculate Enterobacter. So one guy do EA, one guy do EC. Okay, this is your bench partner. Simple. Okay, then we will do a test. You're gonna work together. That's IMVC test. Okay, second test. That will be the last one. Oxidase test. Okay, what is oxidase? We talk about in the lecture. So we had electron transport chain. Okay, we can draw it again. We talk about already. We talk about the cytochroma C, cytochroma A, A3, and finally go to oxygen. Okay, this is the electron transport chain. In bacteria in the plasma membrane system, in eukaryotes at mitochondria, we already talked, and related to the proton motive force. What happened is, for some of the bacteria, once the electron transfer from cytochroma C all the way to oxygen, they need an enzyme. This enzyme called oxidase. And this oxidase, the full name is cytochroma C oxidase. Which means they must use this oxidase, otherwise the electron cannot be finished, the transport. Now how to do it? It's very simple. Left side, inoculate E. coli. Right side, inoculate Citrobacter. They are very important bacteria. Is oxidase test positive? Is Neisseria. However, we cannot do it because of human pathogen. Neisseria meningitis, Neisseria gonorrhea is oxidase test positive. But we cannot do it. We use Citrobacter as a surrogate. And this one, 35 degrees Celsius, 24-hour incubation, and you're going to add a drop oxidase reagent. You don't have to know the name. It's very long. It's about like 25, 30 letters. Very, very long. What we're going to see is after you do the dropping of the reagent, this area should show you little bit blue color. But it's very important, the oxidase test has to do within 30 seconds to 1 minute. If you let it stay there, 1 hour, 2 hours, everything will become positive because there's oxygen in the air. Which means anything will happen. The electron finally transfer. So that's oxidase test. Okay? It is very important used in the clinical area, and we need to know. So these two tests we are to do. Very simple. You inoculate, put the bacteria there. So all these things, did you see what's the characteristics for this biochemistry test? You inoculate the bacteria, you do the confirmation test, and basically you see the color change or the bubble. But you need to know the story behind it. That makes it a little bit difficult. Okay? So that's we finished the biochemistry test. This year, because we have election day to cut off one day, sometimes we have off. So we have to continue to do another test, which is lab 15, antibiotic resistant test. Okay, that's called the Kirby Beau test. So we already know antibiotics, so we're not going to repeat it. That means the metabolic ingredients. I'll just write it, metabolic ingredients. This is basically from fungi to kill or inhibit bacteria. And this one we call it a cyto, and this one we call it a static. Okay, so what is Kirby Beau test then? Kirby Beau test, we need using a very special agar called molar Halton agar. That's MH agar, molar Halton agar. And then you need a solution. We will using just 0.1% nutrient loss. And we need to pick a bacteria. From the slums, we go there and we mix it. Now, how many bacteria this needs to be? There is a standard. This called 0.5 McFarland standard. And 0.5 McFarland standard is approximately 10 to the power of 8 cells per ml. Because otherwise you have 10 to the 5, 10 to the power of 9, 10 to the power of 6, make it inconsistent. So we need to make sure this is the same. Now there is, ideally have a silicon-made standard to compare YouTube and the standards to see the turbidity. I don't have that one anymore because it's difficult to order right now. I just want to let you know. Approximately when you add it into 0.1 nutrient loss is about 0.5 McFarland standard. Approximately 10 to the power of 8 cells per ml. Okay, once you do that, we need using cotton swab. This is cotton swab to put in the solution and do a very gentle streak. A very gentle streak is like this. Very gentle. And then, do not put a cotton swab, come back. No, come back. Do another one. Horizontal one. So vertical and horizontal one, both we do it. After you finish this, this is ready. And you're gonna come here. We have Kirby Beau dispenser. Or press the button, but then the disc will be forth. And I'm gonna only do 2, okay? There are 2 discs right here. So you're gonna get 1 TE30. What this means? Tetracycline. The concentration of the antibiotics is 30 microgram. You may get PEP, penicillin. It's 10, for example. That means it's penicillin. The concentration is 10 microgram. Okay, this guy, we're going to do incubate 35 degrees Celsius 24 hours. What you're looking for is next week, you're looking for the transparent zone. or we hope we have it. You are looking for is this distance. And using ruler to measure. And we will have some table and some standards to tell you. The results will be a different category. So for example, your measuring tetracycline is 15 millimeter. And then you look at your fact sheets and you will see the tetracycline standards is actually this category. So R, I, and S. So for R, we had less than 14. For I, we have 15 to 18. For S, we have 19. What are these stands for? The R represent resistant. S, susceptible. Remember, we don't call sensitive. Sensitive is allergic. People, human being allergic to antibiotics, susceptible. I is intermediate. So if you get a 15, that means it is intermediate. Which means for this specific bacteria, tetracycline, the reaction is intermediate. Certainly you cannot use it. If I have another sample, I get tetracycline, is 21 millimeter, then it's fit this category, which means for this pathogen, tetracycline, 30 microgram for this certain bacteria is susceptible, which means it could be used. But then we need to determine minimal inhibitory concentration. That is twofold serial dilution. Okay, we're not going to do here. This guy, I want to make sure you guys do it easy. I will just suggest everybody just do E. coli, okay? Because we don't have that much antibiotics. I will dispense the two for you. You just do the E. coli, and we will do the incubation, then we'll see the inhibitory result. Okay? So that's basically all we have. Now, I don't need to show you how to do it. It's very simple. 3 agar. MRVP blast. Inoculate your assigned culture. What is your assigned culture? You do E. coli, you bench partner do Enterobacter. So you decided by your bench partner. Okay, we have neutron agar. What we do? Split them onto two. Left side E. coli, right side Citrobacter. And then everybody do E. coli for what we call monohelton agar, using cotton swab to spread on the surface and come to the front desk. We do this dispenser for you. Okay? That's all we have. They also need neutron broth. Yeah.